Bismillahirrahmanirrahim. Welcome back everyone after your summer break. We were at the unit of genetics, and today is your third lecture of genetics. So, don't let the hope die, and we have to survive through it together. So, I will start with the genetic disorders. And when we classify genetic disorders, they are of two types: single gene disorders and chromosomal disorders. In the single gene disorders, they are further divided into autosomal chromosomes and sex chromosome disorders. The autosomal, as we had already studied, can be autosomal dominant or autosomal recessive. And then there are sex chromosome disorders which are sex linked. Mostly they are X linked, but very rarely they can be Y linked as well. And in the chromosomal disorders, the chromosomal anomalies can be numerical or they can be structural, which means there is defect in structure of a chromosome. So here you can see the examples of autosomal dominant disorders include inherited breast cancer, inherited colon cancer, or other dominant disorders like familial hypercholesterolemia, von Willebrand disease, polycystic kidney disease, neurofibromatosis, Huntington's disease, tuberous sclerosis. And in the example of autosomal recessive, you have cystic fibrosis, and then congenital deafness, phenylketonuria, and spinal muscular dystrophy. In X-linked dominant examples, you have Xg blood group or vitamin D resistant rickets, hereditary motor sensory neuropathy, and Rett syndrome. In X-linked recessive, you have example of color blindness, you have example of X-linked learning disabilities, fragile X syndrome, Duchenne muscular dystrophy, and there is very important example which is hemophilia A. And in Y-linked, there is only one example which is XY gonadal dysgenesis. So here we come to the numerical changes of chromosome. That is, there is changes in number of chromosome. As you all know that the normal number of chromosome is 46. However, in case of numerical changes of chromosome, there can be aneuploidy or euploidy. In aneuploidy, there can be hypoploidy, that is reduction in number of chromosome, or hyperploidy. When there is loss of one chromosome, that is 2n minus 1, it can be 45 chromosome. This is monosomy. Hyperploidy, when there is a gain of chromosome. If there is one chromosome which is additional, that is 2n plus 1, it is trisomy. However, if they very rarely there is two chromosome additional, this is tetrasomy, that is 2n plus 2. In euploidy, the multiples are or the number of chromosomes, they increase in multiples. Monoploidy means x number of chromosome. Diploidy means 2n or 2x. Polyploidy means multiplication of 3 times, that is instead of 46 pair of chromosome there can be 69 or more multiples, 4 times or 5 times or 6 times or so on. So here you can see the example of structural chromosomal anomalies. In the previous slide we have done the numerical, that is the change in number of chromosome, and now the structural chromosomal anomalies, which we also known as mutations. So in the A you can see this is deletion, that is loss of a segment of a chromosome. In B there is translocation, that is exchange of chromosomal segments. Here you can see in the pink short one and the blue large one, they both have their lower segments get exchanged here. So but because both of them are balanced in their size, so this is balanced translocation. And then comes the inversion. By inversion you mean that at the point of centromere there are breaks, and the chromosomal break causes the rotation of the chromosome clockwise or anticlockwise. When it is around the center, around the centromere, this is known as pericentric inversion. But when both the breaks are on the same size of his side of his centromere, and they are known as paracentric inversion. And then there is Robertsonian translocation. In the Robertsonian translocation, the short arm of both the chromosomes, they get lost. Here you can see, however, the large arm of both the chromosome, they fuse together. This is known as Robertsonian translocation. And then there is the homologous chromosomes. When they detach during the meiosis, in this case isochromosomal translocation formation, the both the uh, short arms, they fuse together, and both the long arms, they fuse together, so there is the formation of isochromosome. And at the last you can see that when there are breaks in both the arms of a chromosome and they fuse together at their terminal ends, they form a ring. This is known as ring formation, and the fragments which were detached, they are lost. And here you can see that normally in meiosis 1 and in meiosis 2, the chromosomes, they separate and go to all the daughter cells equally. However, in case of non-disjunction, there are unequal distribution and there is unequal detachment of the chromosome. This is known as non-disjunction. And for the understanding of this previous example of structural anomalies, here you can see that the grey cat ran down. This is an original statement. You can take this example as a chromosome example. And in the second one, you can see grey cat ran dawn. As this sentence doesn't make much sense, it is missense mutation. And when grey green cat ran down, here the word green is inserted into this sentence, so this is an example of insertion. And when the cat, this word has been deleted from this sentence, this is known as deletion of a chromosome. And when the some segment of a chromosome is repeated, like grey cat ran down, here you can see this is known as duplication. And at the end there is only one word grey, which doesn't make any sense at all. That means the loss of the whole of rest of a chromosome. This is known as nonsense mutation. And now we will come to the congenital anomalies or genetic disorders, which was our basic topic of today. So first one and the most famous and important one is the Down syndrome, and the karyotype of Down syndrome is trisomy 21, and maternal serum markers or the diagnostic markers of Down syndrome during pregnancy include pregnancy associated placental protein PEAPP, which is reduced, and alpha fetoprotein is also reduced, and HCG is increased in case of Down syndrome. And these are the maternal serum markers, and the fetal marker is knuckle translucency on the ultrasound. And also there furthermore studies say that inhibin A, which is a hormone like substance, it is markedly increased in case of Down syndrome. And now you can see what are the clinical features of a baby with Down syndrome. You can see that there are medial epicanthal folds and slanting eyes, low set ears, depressed nasal bridge, flat occiput, and growth retardation. There is a big protruding tongue, and in the heart there can be congenital malformations and congenital malformations in their intestine as well. And in their bone marrow they have increased propensity of ALL, that is acute lymphoblastic leukemia. And on their hand there is a simian crease, that is a single crease or a single line on their hand. This is known as simian crease. And they have wide gap between their first and the second toe. And then the other one is the Turner syndrome. It is also a very famous syndrome and important as well from the examination point of view, just like the Down syndrome. Here you can see that the karyotype is monosomy 45X. As X, there is a loss of one X chromosome in a female baby, and the diagnosis is clinical, and the treatment is hormone replacement therapy. The female, they have a short stature and they have a thick webbed neck, and on the chest they have broadly spaced nipple, and also they have heart defects including coarctation of aorta and bicuspid aortic valve, and poor development of their breast. And when you see at their elbows, they have a wide carrying angle of their arms. And in case of their internal reproductive organs, there is ovarian dysgenesis, primary amenorrhea, and deficiency of the female reproductive hormones like estrogen and progesterone, and infertility. And also there is retardation of bone age leading to short height or short stature, and lymphedema of hands and feet at birth and later on. Then comes the Klinefelter syndrome. This is 47XXY, that is addition of one chromosome, that is hyperploidy. So here one X chromosome is extra and they are males. These male are a little bit opposite to the Turner syndrome. This is in males, whereas Turner was in females. These males are very much tall, that is tall stature, but they have lack of facial hair and they have narrow shoulders, and their arms and legs are very long. And when you see their blood cells, there is a bar body, which is the remnant of extra chromosome inside the cell. You can see it microscopically, and their testes are atrophied, and there is reduced pubic hair, and they are diagnosed by the presence of bar body, which is a presence of inactive extra X chromosome within the nucleus. They are called super males and have criminal tendency. Then comes the Marfan syndrome. The Marfan syndrome, it is an autosomal syndrome and it is autosomal dominant. There is mutation in FBN1 gene on chromosome 15. This gene encodes fibrillin 1 glycoprotein, which is the main constituent of microfibrils of extracellular matrix, and the loss of this gene can lead to lax connective tissue. So as you can see, the typical Marfanus habitus include very tall male, and they have very long arm span is larger than the height, and they have eye problems, and they have abnormal chest, heart and lung problems, and their abdomen or their torso is short. And signs and symptoms include disproportionately long legs, long arms, toes and fingers, extremely tall and slender build, long narrow face, and high arched neck, and indented or protruded sternum, and their eyes have dislocated lens due to the defect in connective tissue of the lens ligament, and there is a high pressure in the eyes, which is glaucoma, and cystic changes in the lungs. Their joints are flexible and hyperextensible, and their feet are flat. They have curved spine, and their heart sounds are abnormal. So this was the first part of your lecture today. And remember, in the middle of difficulty lies opportunity. So you guys don't need to worry about this pandemic, and we are going to go through this inshallah with safety and security. Then comes the genetic disorders due to environmental influences, which we will discuss in the second part of our lecture.